Hello, everyone. I want to thank you for joining us here today. I'm very happy to introduce this webinar, In Vitro Permeation Testing Applications to Cosmetic Products. So our diffusion webinars so far were mainly focused on pharmaceuticals, but today we're going to learn more on how the cosmetic industry can also benefit from permeation testing and make products that are better and safer to all of us. My name is Bruna Lousada, and I am the European Manager of Teledyne Hanson, and this webinar will be presented by Dr. Hanan Osman-Ponchet. Hanan is the founder and CEO of PKDERM, which is a French biotech company providing innovative in vitro solutions to evaluate the efficacy and safety of pharmaceutical and cosmetic products. So I personally met with Hanan earlier this year at a conference, and I got really excited with her experience in this area. So it's a real pleasure to introduce her. She has a Master of Science in Toxicology and Pharmacology and a PhD in Biochemistry, Cellular and Molecular Biology. She has worked in drug development for over 20 years at companies like Sanofi and Galderma, and with her passion for innovation in skin science and dermatology, she founded PKDERM in 2018. She also authored more than 30 publications and patents and has given presentations at many different scientific conferences. So just before we start, I'd like to mention a few things. Everyone here is on mute, as you will notice. But you can submit your questions anytime by using the Q&A section on the bottom of your screen. And we will do our very best at the conclusion of this presentation to answer all of your questions. If you run out of time, we will email to you directly with the answers. Do not worry. If you're having trouble hearing or if there are any connection problems, we suggest that you log off and log back on. And you should also know that this presentation will be available for replay on the Teledyne Hanson YouTube channel in a few days. With that, I would like to welcome Hanan, and now I'll turn it over to you.
So thank you very much, Bruna, for the kind introduction. I'm very happy to be there in this webinar to present the in vitro permeation testing applied to cosmetic products. And I hope this presentation will be useful for the audience. I will start with a short introduction and then I will describe what is the main or the critical step in experimental procedure for in vitro permeation testing. And finally, I will present two case studies of application of in vitro permeation assay to cosmetic products. So, as you know, absorption of test articles across the stratum corneum can be done through three ways. The transcellular or intracellular intercellular route, and this route is preferred by lipophilic and oil-based ingredients. The second way is the paracellular route, and this is preferred by hydrophilic molecules. And the third route is via the appendage, meaning that the test molecule need to go via the hair follicles and the sweat glands. However, this route is not very common and is preferred by the very large molecules and the molecules soluble in water. What is the difference between conducting in vitro permeation testing in pharmaceutical development and in cosmetic development? In fact, the procedure is almost the same, the experimental procedure. However, the objective when developing a cosmetic product and when developing a pharmaceutical product or a drug is different. For a cosmetic product, we need the ingredient to stay on the superficial layers of the skin and not to reach the plasma in order to avoid any systemic effect. However, when we develop a drug, we need that the drug go deeper in the skin and even reach the bloodstream to have a systemic effect in the case of transdermal patch. So the objective is different. The strategy in developing a drug or a cosmetic product is different, even if the technical procedure to measure dermal absorption is the same. So what is the objective of conducting in vitro permeation testing? The purpose of in vitro permeation testing is to characterize the rate and the extent of transdermal or topical delivery of test items. And we need to answer two main questions. The first one is how the test item is distributed in the skin. It is on the stratum corneum, it is in the epidermis or the dermis. And the other question is, does the test article will penetrate the skin and reach the plasma, and to what extent? So those are the two main questions we need to ask in order to answer. And this is the main objective of conducting in vitro permeation testing. And why we need to have this information in the case of a cosmetic product? In fact, the knowledge of dermal absorption in cosmetic development is essential for safety issues. The difference for a drug, when the knowledge is essential for safety issue, but also, and this is a more important point, for safety, for efficacy issue. In cosmetic, the main focus is the safety, because the quantities absorbed and measured can be taken into consideration in toxicological risk assessment in order to extrapolate the human exposure and to calculate the margin of safety. So this assay, the in vitro permeation testing, is applied for different purposes in cosmetic development. We can use this assay in order to screen and to select the cosmetic ingredients with expected penetration properties, to characterize the skin distribution of cosmetic ingredients, for formulation screening, and the most objective is to for safety assessment, as I said before. In terms of regulation or regulatory guidelines, there is many documents, regulatory documents that that describes the rules and how to conduct in vitro permeation testing. In cosmetic development, we use the OECD test guideline 428 for skin absorption in vitro method, and the Scientific Committee on Consumer Safety regarding the basic criteria for the in vitro assessment of dermal absorption of cosmetic ingredients. And as I said, there's other documents recommended by the EMA, US EPA, the WHO, that are related to dermal absorption. Now, regarding the experimental procedure of conducting IVPT, this assay is conducted using, as you know, the famous France diffusion cells. So what is the France diffusion cell? It is composed of two compartments, the donor compartment and the receptor compartment. And the skin or the membrane is placed between the both chambers, and the test product is applied on the surface of the skin. And the cells, the diffusion cells, are normally incubated at 32 degrees under continuous mixing. As you can see here, there is a mixer. There is a possibility to collect the sample of receptor liquid at several times in order to make a kinetics of the absorption. We can use different types of formulation. We can use liquid formulation or semi-solid as creams, gels, foams, lotions, or ointments. We use in this assay human skin as a gold standard. Normally, excised human skin comes from plastic surgeries. And in our lab, we use skin from abdomen area as they constitute a very large samples. In this assay, we can use both fresh or frozen skin samples. When we are interested to evaluate the metabolism of drug transporters, we need to use fresh tissues in this case. And regarding the number of donors, when we evaluate the cosmetic products, the SCCS recommends the use of at least 8 skin samples from at least 4 donors. We need to perform a strict quality control when performing IVPT. We should control or check the thickness of the skin. And then, sorry, in this assay, there's a recommendation to use split thickness skin, because the maximum thickness should be no more than 1 millimeter. So we need to measure skin thickness of each sample. We need to ensure also the skin barrier integrity by measuring the transepidermal water loss. Regarding the treatment duration, in fact, it depends on the clinical use, on the condition of use of the product. You can treat for 5 minutes till 24 hours, depending on if you put only the product 5 minutes like a shampoo and then you wash, or if it is a skincare, you apply it every day. And regarding the application amount, we use a finite dose between 5 and 10 milligrams per square centimeter, as recommended by the guidelines. One of the critical point of this assay is the choice of the receptor liquid. In fact, we need to ensure that the test article is soluble and stable in the receptor liquid, and you need to ensure the skin conditions. Usually, we use a buffer like a PBS with a surfactant to increase the solubility. And we perform a solubility assay before doing the study, and also a stability assay before doing the main study, the main experiment. Regarding the analysis of samples at the end of the treatment period, so we can analyze the product in different compartments. Of course, in the receptor liquid that give idea on the absorbed dose. We can analyze in the total skin, including dermis, epidermis, and stratum corneum, or in different compartments, in different layers of skin, like dermis separated from epidermis, separated from stratum corneum. And also we analyze the compound in the excess of formulation and the washing when we need to calculate a mass balance at the end of the study. Regarding the analytical method now, different analytical methods can be used to quantify the concentration of test item in the different skin compartments. LC-MS is the most important method that can be used. For some ingredient, you can use ICP-MS or LCUV, or even using radio. If the ingredient of the test article is radio labeled, you can use liquid scintillating counting. Sometimes, and it is important to have to visualize the compound in the skin, and in this case, we can use fluorescence microscopy if the compound is fluorescent, or in the case of radio-labeled compound, you can use autoradiography, and you can see really the distribution in the different layer of the skin. And we can use also the multi-mass spectrometry imaging that give both qualitative and quantitative measurement of the distribution of the compound with a different layer of the skin. Regarding the LC-MS, as I said, this is one of the method, the most used method in this assay. Why we use LC-MS? Because we need a very sensitive and accurate analytical method. With generally the limit of quantification is about 1 nanogram ml when possible, because sometimes it is difficult to reach this kind of sensitivity. And validation of analytical method is done in every matrix in receptor liquid, stratum corneum, epidermis, dermis, and washing. So at the end, we have a huge amount of samples with different analytical methods did to validate before doing the study. And this is why this kind of study, the IVPT, is a very expensive study. Now I'm going to share with you two case studies. related to cosmetic product. The first one is we conducted this study because the objective was to quantify the dermal absorption of the cosmetic ingredient, and I named it X on ex vivo human skin. So here you have the experimental conditions. The study was a GLP-1 because the result of the study were needed for the dossier registration. We use split thickness human skin samples, frozen skin. We use samples from 4 donors from abdomen area, and each condition was performed in triplicate, giving 12 samples per treatment condition. Regarding the diffusion cells, we have a diffusion cell with 2 square centimeter surface area, with 7 ml of the volume of receptor compartment. And regarding the receptor fluid, we use PBS plus 0.1% of Twin 80 as surfactant. Regarding the quality control, we measure skin thickness and the TWL to check the integrity of the stratum corneum. In terms of treatment conditions, we were in static conditions. We applied 5 milligrams per square centimeter of the product. We treated for 24 hours with sampling of receptor liquid at 30 minutes for 6, 8, 12, and 24 hours. Analysis of the test article was done using validated LCM-SMS method with a limit of quantification of 1 nanogram ml and generalized in all the compartments of the assay. So here you can see the result of mass balance. So mass balance is the sum of the product in every component of the assay. And we can see that the mean mass balance is within the acceptance criteria validating the result of the study. So measuring the mass balance is mandatory for a regulatory study, because if you are not in the acceptance criteria, that means you invalidate your result. And here you can see the result of quantitative analysis. You can see here that we have a classical distribution profile of the product with the concentration gradient between the outer layer of the skin, the stratum corneum, followed by epidermis, dermis, and then the receptor liquid. So this is really a classical distribution profile. Sometimes we have some exception. But what we can see, how we can interpret the result is the test item X is distributed in the stratum corneum, in the epidermis, and the dermis, and to a lesser extent in the receptor liquid. And as we performed a collection of the receptor liquid, so we can do a kinetic profile and we can measure the absorbed dose. And you can see that the absorbed dose increase with increasing the treatment time, with an average of the dose of the absorbed dose is about 0.7% of the applied dose. And the data of this IVPT were used in toxicological risk assessment to predict human exposure, and the data were included in the registration dossier of the product. Now the other example is to compare or to measure the in vitro dermal absorption of a cosmetic ingredient formulated in 3 different products, 3 different formulations for toxicological risk assessment purpose. In this study, we had 3 formulations, formulation 1, 2, 3, with different concentration of the test item. And in this study, we use ex vivo skin samples, full thickness in this case, and we use 3 human donors and 3 replicates. We treated for 24 hours, and we analyzed in the test item in receptor liquid and in total skin. In this case, we didn't separate each compartment of the skin. And analysis has been performed using LC-MS method. So the comparison of the 3 formulations, we can see that the concentration of the cosmetic ingredient penetrated in the skin is proportional to the concentration of in the formulation. This concentration in this study did not exceed 10% of the applied dose. And as you can see, the formulation 2 and 3 are quite equivalent in terms of the dermal penetration, even if the concentration, as you can see, were quite equivalent. However, for formulation 1 with 30%, we have much more dermal absorption of the compound. So dermal absorption data of this study were used in toxicological risk assessment to predict human exposure. So to summarize, in vitro permeation testing or in vitro dermal absorption is a critical tool to understand delivery of cosmetic ingredients into the various layers of skin. This assay is very important for safety assessment of cosmetic product, and we saw that we need to be careful when we conduct IVPT studies. It is important to ask the question at the beginning and before designing the experiment. And we saw that what is important is the quality control, to check, to be sure that the result you will have are very accurate. And finally, I would like to thank you very much for your listening, and please ask your question or send an email. Thank you very much.
Thank you so much, Hannah, for this excellent presentation. I'm sure it was very useful and interesting to the audience. So now, as Hannah said, we will open for questions. So just a reminder for everyone, you can submit your questions using the Q&A button at the bottom of your screen. Okay, so here's the first question for you, Hannah. In your experience, which are the percentages of penetration in the stratum corneum, epidermis, and dermis that you usually obtain from cosmetics?
In fact, can you hear me?
Yes.
Yes, in fact, it depends on the molecule you are testing. What we see normally is a gradient of penetration, meaning we have a higher concentration in the stratum corneum than in the epidermis and dermis, like in the example I showed. But to really answer this question, it depends on the molecule you test. Sometimes it is difficult for some components or for some compounds to penetrate the stratum corneum, and sometimes you have example where it is not so difficult to penetrate. So it is based on each case is different.
Okay, thank you. Next question. There are many great questions that they don't stop popping up here for me. So we'll try to go over all of them. If we're not able to get back to you, we will send you the replies via email.
Yeah.
So a couple of questions. What is the storage time of frozen skin samples?
Normally, we can maintain a skin sample frozen for 1 year.
1 year.
That is the experience we have. We have procedure for that, and there are studies showing that within 1 year, skin sample can be of good quality, but not more than 1 year.
Okay. How do you achieve a finite dose application?
The finite dose application recommended by the regulatory guidance is of 2 milligrams per square centimeter, but it is difficult to use this amount. So we applied between 5 and 10 milligrams per square centimeter, and this we can weight the amount we applied on the skin in order to get quantitative measurement, accurate quantitative measurement and application.
Perfect. When the skin samples are frozen, do we need to make a treatment before putting them in the front cell?
No, we just saw the skin sample before putting them on diffusion cells, on front cells. So you need to wait about 30 minutes to thaw and to get room temperature.
Okay, about 30 minutes.
Yeah.
Perfect. Can you explain the procedure that you use to separate and quantitate, which is quantify the drug in each layer of the skin? I think it means like stratum, corneum, dermis, epidermis.
Yeah, so it is a long procedure. I think we need to to do another webinar just to show how we can proceed, because it is not so easy. There's a lot of precaution to take when we separate each compartment. We need to avoid any contamination between the samples and between each compartment. Yeah,
Okay,
I think
we
need.
Yeah, maybe tape stripping for the stratum corneum.
Yeah,
and then,
absolutely,
yeah.
absolutely. We make a tape stripping to separate the stratum corneum. We collect the strips, and then using tweezer and scalpel, we can separate the epidermis and collect the epidermis in another tube, and also the dermis. And then regarding epidermis and dermis, we need to crush the samples in a special machine to get better extraction of the test item we need to analyze.
Perfect. Next question. About the receptor fluid containing PBS. So does the molar concentration of the PBS is important for the diffusion studies, or can we use classic PBS?
We normally use a classic PBS.
Okay. Why was the pH of the receptor median in the first case study 7.2, given the fact that the pH of the skin is around 5.5?
Yeah, in fact, the pH of the skin is around 5. It is the surface of the skin. However, the skin is in contact with systemic circulation, and it is at physiologic pH 7.2. So this is why we mimic the pH of PBS to mimic the systemic circulation.
Perfect. Next question, how do you ensure a uniform thickness of the skin across the skin donor models?
I know the question is how to prepare skin sample of about 500 micrometer. In fact, we use what we call a dermatome to cut the skin to get a precise thickness, and we measure in fact each the thickness of each skin sample, and we normally get homogeneous thickness. So we use a dermatome. This is the machine. Yeah.
Okay. Another one, what is the recommended transepidermal water loss to ensure accurate results for common cosmetic products?
I think this depends on each lab. You need to have historical data to get your reference of TWL, because it depends on the machine you use to measure the TWL. In our lab, for example, we set the acceptance criteria about 9. Because we saw that it is the average of the measurement we got for more than 100 or 1,000 samples we measure. But I think it really depends on the lab. It depends on the machine you use, and you have to set up your historical data to define what is your acceptance criteria for TWL.
Very good. Another one, is it desired for the cosmetic ingredient to permeate through the skin or not?
Yeah, this is the question that makes some confusion. Because in fact, normally in our hand, a cosmetic product have to not penetrate the skin, because the action or the effect is on the superficial layer of the skin. However, in fact, it depends on what data we have on the product. If, for example, we have enough data showing that there is no problem in terms of systemic effect, if you have toxicological data showing that even if it is absorbed, there is no adverse effect or something like that, there is no problem. So it depends on what we have, what we know about the product. So this is why, in fact, we perform IVPT in order to evaluate the absorbed dose and to predict the dermal penetration and in human, and this is taken into account to calculate the safety margin. So if the product it penetrates but the safety margin is so high, there is no problem.
Very good. I see there are many questions, so there are many people interested to understand the techniques and the procedures that you use to separate the stratum corneus, epidermis, dermis. I think you already explained, but we can probably answer everyone of those other questions via email if you have a procedure that you can share. But
and if they are interested, I will be very happy to organize a webinar to explain step by step with some video, with some picture, because in fact it's still not it's still abstract if we never see how we can perform this technique.
Perfect. Yeah, that would be brilliant. There's enough content to put another webinar together just for this.
Absolutely.
Okay, that's good. There's a few more. I'll probably reply two more. So how difficult are IVPT studies with very hydrophobic substances, and how do you run IVPT with ingredients that are complex mixtures, UVCBs?
Yeah, with the complex mixture, the question is to define what is the component we will looking at, because if you have a mixture of component and all the components don't have the same behavior in terms of dermal absorption, so this is a bit difficult to select which compound you will analyze, because for each compound in the mixture you need to quantify the absorption. So this is something I'm not really familiar with.
Okay. You mentioned you use skin from abdomen, but skin is this part of the body is different from in your hands, your legs, your feet, or your face. How could you generalize the results from the abdomen skin that you obtain to other part of the skin from different parts of the body?
Yes, this is a good question. However, if you want to perform dermal absorption, for example, on the leg or on the face, it is difficult to get the samples, because to perform that, because we need a huge amount of skin sample to perform this kind of study. But now I think you need to have a look on the result in general, and then you can put a corrective factor in terms of the region you need to treat, in fact.
Okay. Continuing the membrane in the skin, is it possible to replace the human skin with other types for preliminary tests, like any type of synthetic membrane or pig skin?
Yeah, pig skin is used with many people use pig skin instead of human skin, because it is not so easy to get human skin, and the study shows that pig skin is relatively similar to human skin, so you can perform a study with pig skin. Now, when you work with cosmetic product in terms of image and in terms of marketing, if you want to communicate, I think for me and my client prefer to work with human skin. Just in terms of
the gold standard, as you said, presentation. Okay. One last one, and then we'll finish for today. So how do you guarantee that all stratum corneum is removed by tape stripping? Is there any way we can guarantee?
Yeah, in fact, you cannot guarantee because you have when you normally we perform tape stripping till we cannot remove anything, and then we stop. But sometimes you have the stratum corneum in the furrow of the skin, and this part you cannot remove it. So you have to take into account when you analyze epidermis, you have some contamination with the stratum corneum.
Brilliant.
Yeah.
Okay, so thank you so much, Hannah. And I think that is the end of today's webinar. Many great questions. Thanks for sharing your knowledge and your experience with us. And of course, thank you everyone who attended the webinar, everyone who interacted with us and sent all those great questions. If we didn't get back to you today or now, we will reply back via email, so don't need to worry about that. As I mentioned, there's a lot of questions also about if this will be available, if we can send it via email. As I mentioned earlier, the webinar will be on the Teledyne Hanson YouTube channel, probably within a few days, and you'll be able to watch this on demand. And please feel free to share with people that you think could also benefit from this content. So thank you very much, and I hope to see you in our next webinar, maybe about the skin preparation, because there was a lot of questions about how to do the separation of the skin.
Yes,
so,
thank you very much. Thank you.
Thanks, Hannah. Thank you. Thanks, everyone.